In this chapter, we will explain the data collection, digitization, cleaning, and database storage processes of 150,000 people's cohort study data extracted from the participants of the cohort study. The data collected in the cohort study of the Tohoku Medical Megabank project can be roughly divided into 6 types: specimen test data, physiological test data, sphygmomanometer and pedometer data, questionnaire data, municipality-provided data, and clinical data. First is the specimen test data. Immediately after collection, the blood and urine samples are entrusted to the testing company, and in a few days, the results are obtained as electronic data. The data is immediately stored in the health information system. The system manages the information related to cohort study based on the cohort ID. Data related to cohort study, such as the current stage of the participants' biological specimens and questionnaires, whether the data is already stored, and if stored, what their values are, and so on, can be consolidated and managed. The next is the physiological test data. Data of the respiratory function test, body composition, and other related physiological tests conducted at each community support center are stored in the health information system through the equipment coordination system. Similarly, the data obtained from the questionnaires conducted at centers using a tablet are also stored in the same manner. The sphygmomanometer and pedometer data are measured by the participants themselves at home for a period of 10 to 14 days. Since data are recorded in the main part of the equipment, the data are taken out at each center and stored in the health information system after being received by courier. The data are handled in the same manner as the physiological test data. The next is the questionnaire data. All the paper questionnaires are collected in the headquarters once and digitized by specialized vendors. Once the data are delivered, they are stored in the health information system. The data of the questionnaire conducted at centers through the tablet is stored immediately using the equipment coordination system. Further, in some follow-up surveys, the data entered through the web are stored immediately after responding to the questionnaire. The next is the municipality-provided data. Data obtained from specific health checkup that is organized by municipalities is delivered as electronic data, then stored in the health information system. It is important to understand the kinds of illness that participants of the cohort study develop later. In the survey, we request that medical institutions provide clinical data based on the report of the follow-up questionnaire. The clinical data necessary for the research is de-identified and stored in the supercomputer system. In order to efficiently acquire clinical data on large scale, it is important to digitize and network the medical records of each hospital. Due to the experience of losing many medical records affected by the Great East Japan earthquake, the construction of disaster-resistant medical information network is underway. We are planning to coordinate with the Miyagi Medical and Welfare Information Network, known as MMWIN, promoted in Miyagi Prefecture, which will be useful for our future surveys. Cleaning is performed at each specific interval of data. Cleaning is a process of checking whether the obtained data show errors during digitization and whether the data are statistically valid and logically consistent. In some cases, this process includes going back to the original data to make corrections and notes. For example, despite selecting non-smoking on the questionnaire, the number of cigarettes smoked is entered. The errors are checked and modified during digitization, and comment indicating inconsistency is added. The physiological test data may vary depending on factors such as the season, test environment, and difference in person conducting the measurement. Hence, data are monitored. Stable data are obtained by conducting maintenance and replacement of the test equipment as necessary and revalidating the measurement methods. After the completion of cleaning, the data from A to F are sequentially anonymized. Data such as genome analysis data and omics analysis data that is obtained from blood and urine analysis are integrated into the database called DBTMM after anonymization. Enormous amounts of data of various kinds, such as data from specimen tests, physiological tests, questionnaires, clinical data, and various analyses are integrated into the supercomputer system and stored in the DBTMM. The DBTMM that centrally manages a wide variety of information is available for various analytical uses, such as distribution of biological specimens and information to external research institutes.